Welcome to Study Time, a televised home learning program produced by Rwanda Education Board.
Hello students, welcome to the TV learning series. As you know, we're going to be studying biology lesson today. I'm teacher Hurbert again. We'll be taking you through the biology lesson of today. So students, today, the biology we're going to be studying, we're going to be studying what you call a practical number one, which is called food testing. Today, we're going to be discussing about testing for starch, testing for proteins, reducing sugars, and I said reducing sugars, examples are glucose, we have galactose, and fructose are examples of reducing sugars. But we're going to use the glucose as an example of reducing sugars. We're also going to test what you call non-reducing sugars. And examples of non-reducing sugars, we have the sucrose. Add here the word sucrose. We're going to be testing fats. We can extract fats in very many things. We can use cooking oil. We can use nuts and etc. We're going to be testing vitamin C, which is ascorbic acid. And as well, we're going to be using distilled water. Why, students, are we going to be using distilled water? The reason why you use distilled water, it is going to be using as a control experiment. If I say a control experiment, it's an experiment that tests negatively. So, students, here are the reagents and chemicals that we're going to be using today in testing the food components. We're going to be using iodine solution or iodine reagent. We are going to be using bendic solution. We are going to be using burette reagent or Merion's reagent or copper to sulfate. Because if you have three of them, they can just be used for testing the presence of proteins in the food. We have the ethanol, which is very important in testing what you call fats. That's like we're going to use examples like cooking oil. We have DCPI, which is endrophenyl dichlorphenyl, which is going to be used for testing vitamin C. So students, today, as we're going to start our practicals, let us have some chemicals, the names of the chemicals and the names of the food that we're going to be using to present our lesson today. I have very many things in front of me. Very beautiful things in front of me. You can see in front of me, I have a very well-decorated table, having different food compounds, having something like reagents. You can see and observe. I'm going to be discussing with you how to make a test about these food compounds. Very good. You've seen that I've mentioned about the foods that we're going to be testing or the nutrients that we're going to be testing. I mentioned about the chemicals that we are going to be using as far as testing, as food test is concerned. And there, you can see I'm standing in front of a table and which is well decorated. There are very many food compounds that are in front of me. And there are very many chemicals that we're going to be using for testing these food compounds. As well as I'm going to start with table one, what is in front of me. I will show you. And I think everybody knows what is this. We can say, this is a sweet potato. And I can say in sweet potato, I can extract starch from there. And I test the presence of starch. I can use cassava. I can use Irish potato. This contains starch. I even remember I can also use rice. I have something which is also a laboratory prepared starch. This is a prepared starch. But it is extracted from plant tissues. Table 2, on a preparation, sample 2, this is sample 1. This is sample 2. Sample 2, I have something very interesting. What is this? An egg. What does egg contain? We normally say the egg is made up of the yolk, the egg albumin. So I'm going to extract the egg albumin and I use it to test the presence of protein in the egg. I have some beans, cooked beans. As you can see, these are cooked beans. These are cooked beans. And I'm going to use them also to show that these beans contains proteins. I have milk. I think everybody takes milk. What you call it is a bodybuilding food. Lack of milk suffer from kwashiorkor. I have sample C. In sample C, I have something very interesting, very beautiful. I can see, you can see, and you observe. What is this? Biscuits. I'm going to use glucose biscuits to test the presence of glucose. If I say glucose, I mean reducing sugars. Examples of reducing sugars are monosaccharides. I have sample D. Sample D is made up of non-reducing sugars, which is sucrose. Sample C is made up of ascorbic acid or vitamin C. We can also extract vitamin C from lemons. We can extract from lemons, from these fruits. I can say fruits, in all fruits, you can extract vitamin C. I have sample F, which is made up of cooking oil. I'm going to test the presence of fats in cooking oil. I have sample G. If I can see sample G, distilled water. If I say distilled water, it means it is a water without any impurities. So, and I can ask you, why do I have to use a distilled water? I use a distilled water acting as a control experiment. If I can say a control experiment is an experiment that shows a results in a negative way, not in a positive way. All right? So I have some chemicals in front of me. The brown one, the blue one, the colorless one. I start with chemical 1. I have iodine solution. I have solution 2, which is burette reagent or using for testing of proteins. In testing of proteins, you can also use Meron's reagent. You can use Meron's reagent to test proteins. Also, remember, we can also use copper 2 sulfate to test the presence of proteins. I have a chemical 4, which is Benedict solution, testing of reducing sugars. If I say reducing sugars, I mean glucose. Excellent. I have another chemical here, which is hydrochloric acid. I have another chemical, which is called sodium hydroxide, a base. This one is an acid, and this one is a base. I have another solution, which is called dichlorphenyl and rofenyl. Dichlorphenyl and rofenyl. Excellent. We have another solution, which is the last solution. We have ethanol. What does ethanol do? The ethanol is used for testing for testing fats. All right, student. Excellent. We have seen all the reagents we are going to use, all the food we are going to be testing. Let us now start our practical of testing starch. What do we use in testing starch? We said testing starch, we use the iodine solution. Let's start our practical. I will have 2 test tubes. I will try to use that 2 test tubes to test starch. I will get a drop of, I can say 2 drops of starch. Or then, after getting 2 drops of starch, I add the iodine solution, and then I will be able to observe color changing. Color changing. Once you see there's a color change, from one color to another color, we say there is presence of starch in the solution. But if the color does not change and persists, we normally say there is absence of starch in the solution. Let us do the experiment. I will use the droppers. This is the starch. We have to stir. As I mentioned, this is starch solution, a prepared starch solution. So you get almost like 2 drops of starch, you put them here. And this is test tube 1. Test tube 2, you put water, distilled water, which is going to be acting as a control experiment. Now, we are going to add the iodine solution. We have test tube holders. Let us get test tube holders, and we try to observe clearly. I'm going to add, I said, what do you use to test starch? I normally use what you call iodine solution. You pour the iodine solution into a beaker. After adding into a beaker, you get the droppers. After getting the droppers, and then you try to see if there is color changing. You add the same amount as the amount of the starch you have added. You try to observe 2 test tubes. As you can see, I have 2 test tubes. I have test tube on the right-hand side. I have another one in the left-hand side. Right, left. I said test tube 1, I put there water. Test tube 2, I put there what you call starch. Student, from there, from these ones, you can make your observation. The observation is that test tube, I can say this one in the right-hand side, it doesn't change. The color of iodine does not change. It persists. So meaning that this solution does not contain starch. But this one, as you can try to see, now when it changes, the positive result shows that there is presence of starch because the solution has changed from the brown color to a blue-black color, which indicates the presence of starch. Thank you. Okay. Now, we are going to test for, so I'm going to use that one to test about the presence of starch. Will that one contain starch? I will use my knife. As you can see, I will use a knife. I extract a small, just get a smaller piece of it. I put it into a mortar and a paste. I try to crush it to see if they have the same results. This one was a prepared starch. This is the starch I'm extracting from the sweet potato. This one is, I'm using a mortar and a paste to crush the food. Okay? Now from there, I'm going to use the knife and I put it into a test tube and I see, do these contain starch? Okay. From here, I'm going to put there the iodine solution. Let me first get another test tube and I put there the distilled water. The distilled water and I test if it contains. Here I put here the distilled water to act as a control experiment. Do the sweet potato contain starch? Now, I will try to make it and I make it, you observe it and tell me if this one is acting as a control experiment. I'm going to put the same amount of iodine solution now. in the two test tubes. I add the same drops. From here, you can make the observation now. Here, solution this one, the solution this one does not contain starch because the color of iodine persists. But this one, you can see the color of iodine has changed from a brown color to a blue-black color, which indicates that in sweet potato there is presence of starch. There is presence of starch. Here there is absence of starch. Here there is presence of starch because the color has changed from iodine solution, the color of iodine solution has changed from brown to blue-black. Thank you. Take a note of this. Write this one on your book. Take a note. You can say, take a note, I think. You say, add iodine solution. You can say 2 centimeters cubed or 2 drops of iodine solution. To starch the iodine, which is brown in color, turned into black. Indicates presence of starch. You can say this is positive. Then when the color of iodine persists, does not change, it means absent of starch. Number 2, we're going to test about proteins. What do we use to test proteins? I will use, again, the same apparatuses. I will use 2 test tubes. You see the test tubes? The test tubes are going to get 1 test tube I will put there. I will put here water, which is acting as a control experiment, which tests negatively. And this one, if I put here something like a protein, I will also try to observe and see. Let us try to do it and see. I will put water in 1 test tube, which is a sample G. Then, and that's another test tube, I will put there a protein. I will use the protein to see if there is presence of starch, presence of proteins. Here, this is an egg albumin. I extracted the egg albumin from here. Now let us see if it contains proteins. The same amount. I'm going to use a solution here which is called testing. I'm going to use the burette test. The burette test. Good. Students, before I use the Burette test, you can see the color of the Burette solution or Burette reagent. It is blue in color. If the color of the Burette persists, it means the sample does not contain proteins. But if the color of the Burette changes from blue to purple, that one indicates the presence of proteins. Let us try to use the pipette, then we add them into 2 test tubes. We start with that one. Use like 2 drops. Then shake gently, and you try to observe the color changing. You see the difference now from here? You see the difference? Here, the color of Burette persists, does not change. But here, the color changes from a blue color to a purple color. If it changes to a purple color, that one indicates there is presence of proteins. So proteins, the color that shows that there is presence of proteins, the color changes from a blue color to a purple color, which indicates positive. The positive result shows that there is presence of proteins. Good. Students, how we have seen that the egg whites contains proteins, let us also test if the milk contains also proteins. Let us try to make it practically. Let us get the 2 test tubes. You can see these 2 test tubes, they are empty. We're going to put water in 1 test tube. I'm going to get the next test tube. I'm going to put there milk. And I told you that we normally use the Burette test or the Burette reagent to test the presence of proteins. Let us put the reagent and we observe the color changes. If you observe the color changes, we make a deduction or a conclusion. Let us put the reagent. In the milk, I add 2 drops of Burette solution. And then I add in water Burette test, testing for proteins. We shake the 2 test tubes. I think from here we can make an observation. Then from making the observation, we make a deduction or a conclusion. We can say test tube in the right-hand side does not contain protein because the color of the Burette persists, doesn't change. But here we see the color of the milk is white in color, but the color of the milk is changing from white to purple color. If it changes to purple color, that one indicates that there is presence of proteins. So in conclusion, we can conclude by saying that in the milk, there is presence of proteins. In water, there is absence of proteins. Good. So, students, we thank you for studying and active participation. I will come you to the next lesson of practical biology. We're going to make a practical concerning about ascorbic acids, which is vitamin C, cooking oil, non-reducing sugars, and reducing sugars. Thank you so much for being a part of biology lesson. See you next time.